Hi, welcome to our Lab 6 presentation for physio. Today, Ben Francis, Audrey Kim, Jessica Liu, and I, Daniel Wall, will be giving a presentation of our experiment, which we tested reaction speed on the type of color that we saw.
Okay, so first our hypothesis. Our hypothesis was that the reaction times would be faster for brighter colors, so in this case, light green, than darker colors than regular green or dark green. As you can see, for the dark green, we predicted an increased reaction time when compared to the light green, and we based our hypothesis off of the study that we listed here. And in this study, they found that brighter colors, which weren't just the greens that we used, but brighter colors in general, had a decreased reaction time, and darker colors had an increased reaction time. So we can see that with the arrows that we've indicated in our figure. And we based our hypothesis off of that study and our own rationale, which I will discuss in the next slide. In these two diagrams we see on the bottom, these are basically two diagrams of how we see. And on the left, we have the mechanism of how we see. So we see that light is picked up by the photoreceptors, which is transferred into an action potential, and it's sent through bipolar cells and which is sent to ganglion cells. And then finally, that action potential is sent to the brain for processing. And on the right, we see just a cross-section of a rod, which are the light-sensitive photoreceptors, as opposed to cones, which are the color-sensitive ones. So rationale is that in lighter situations, more rods will be activated. And when more rods are activated, this will cause an increase in signal strength. And as we know, when there's an increase in signal strength, the activation threshold will be reached quicker. And this is what will cause us to have a decreased reaction time. And the reason why we hypothesize that light green will have a smaller reaction time than dark green.
Thanks, Ben. So for the method of our lab, it begins with each subject accessing the link attached. This link is a test to find out how fast the subjects respond to different colors. And as mentioned before, the colors we're focusing on are lime green and dark green. So group 1, which was Jessica and Daniel, set their first color to lime green. And group 2, which was myself and Ben, set their color to dark green. We chose to have our groups begin our trials on different colors as a means to control for exposure to the first color. And one challenge we had was taking this test under different conditions because we're all in different locations. So we also made sure that each subject was relaxed before taking this test, with their back straight and at eye level with the screen. This test begins with pressing the start button and waiting for the background color of the monitor to change. And once the color changes, the subject presses stop. And the reaction time is given on the screen, and then the subject records their data into a table. And steps C through F are repeated a total of 9 times before switching colors to the opposite brightness, depending on your group. And then steps C through F are repeated another 9 times.
So for our results, we have set up a table here that shows the average reaction time for our lime green, the average reaction time for the dark green or normal green, and then the p-value. So Ben, Audrey, Jessica, and I all had a higher average reaction time for the darker green than we did for the lighter green. As far as our standard deviation goes, our spread was rather small, which is a good sign for consistency. We then ran a t-test to show which reaction times or the differences between our reaction times are significant. And we found that every single one of our p-values was lower than 0.05. which lets us reject the null hypothesis that the differences between our reaction times was due to random chance. And so on the next slide, we see here is a graph that basically just uh, shows the table, but in a image form. And on the left, we have reaction time in seconds, which is why they're in decimals here. And on the x-axis, we have the participants, so Ben, Audrey, Jessica, and I. And then we had each person had a different reaction time measurements for uh, each color. So Ben's green, Ben's lime, Audrey's green, Audrey's lime, Jessica's green, Jessica's lime, and my green and my lime. And as just to explain this x-axis here, the green represents dark green, the lime represents light green. And as we see here, all the averages, as stated earlier, are lower for the light green, which represents a faster reaction time or reaction speed. And as for the error bars, we set up the error bars to be the standard deviation. And as we see, we have a relatively small spread with Jessica and I maybe having a slightly larger spread than what we would prefer in the future. But this might have to do with our less than ideal trial size, but in the future, we might want to up the amount of trials that we do so that this spread decreases and that we get more data that is not as spread out and more reliable.
So going back to our hypothesis that reaction times will be faster for brighter colors because it would activate more photoreceptors, more specifically rods, I will be talking about how light is processed. Light first must process by passing through the cell layers of the retina before reaching the photoreceptors. The rods and cones that make up the photoreceptors differ in their shape, range of wavelength, and function. The rods contain rhodopsin, and the rods lack spatial resolution and color function. The rods usually contain the rhodopsin in a membrane-bound disc, and the rods lack spatial resolution and color, which will cause it to function well in low light environments and allow the movement of peripheral vision. And the cones contain opsin, which humans have 3 types of cones, which are red, green, and blue cones. The cones have high acuity, low sensitivity, spatial resolution, and good color perception. The cones are sensitive to different wavelengths of light, which allow us to have color vision. The relative activation of red, green, and blue cones allow us to perceive different colors. And in bright conditions, cones allow us to have good color vision. However, in darker conditions, the cones aren't as active as the rods. And after the light reaches the photoreceptor will release different amounts of glutamate onto bipolar cells, depending on the light intensity. The bipolar cells then synapse onto ganglion cells that fire action potential to the brain. The firing of action potential encodes information about the light. And on the figure here shows the different types of cells that make up the retina and the organization of the cells. So for future plans, one of our future plans would be use different colors to allow us to see if activating different cones would increase or decrease reaction time. This factor could affect the reaction time because different colors have different wavelengths. And the second would be changing the environment while we're taking this test. By changing an environment from light to dark, it could affect the rods in our eyes, thus affecting the reaction time. And lastly, comparing reaction time in the morning, afternoon, evening, or night could be a factor because the reaction time could be correlated with how tired we are. Thank you all for listening to our presentation.